Hello, everyone, and welcome. This is our IVF webinar once again. I hope you had a wonderful weekend, and again, glad to be back with Dr. Uliana Dorofoyeva. You can see her right here. Hello, Dr. Uliana. How are you feeling tonight?
Hello, Carolina, and hello, everybody. I'm great. Thank you, and happy to join again.
Thank you so much for joining us. For sure, once again, I'm not sure even which webinar it is, but I'm very happy that you are here and you are going to present this particular topic because this is a topic that brings lots and lots of questions. And I'm sure that you find this useful. As always, Dr. Uliana has prepared a presentation with lots and lots of details that you will find interesting, I'm sure. And of course, afterwards, she will be happy to answer all of your questions. And we will discuss, of course, egg donation, genetic inheritance. And of course, she will answer the question that I'm sure you ask a lot. So, will the baby look like me? Okay. So, of course, if you'll have any other questions, you can go ahead and type those in the chat section during the presentation or afterwards. And Dr. Uliana, I'm sure, will be able to help with that. And let me just mention, if you don't know yet, Dr. Uliana, she's the medical director at International Corporation at IVMed, which is located in Kiev, in Ukraine. But she's also a medical director at Ovugene Egg Bank, also located in Ukraine. So it's wonderful to have you back. And well, I guess that is it from me at this point. Let's not waste time and let's get to our topic tonight. Okay, Dr. Liana?
Yes, absolutely.
Excellent. Thank you so much. And let's start with the presentation.
Thank you, Carolina, for a great introduction. And dear patients, you know that oocyte donation for me is the most important part of our activities, of myself and our team. And let's talk about the topic, which is very interesting for the patient. And this is true that many patients are asking the questions, how the baby look like once I'm using oocyte donation, and what is important to consider once planning this. So we all know that we all are different, as well as our children are, or they will be. But before we will start moving into the fact of creation of the embryo, let's start from the cells, which are the strategic cells for the embryo creation and the future child presence. So we need to talk about human oocyte. Either this will come from the mother, because we have those options. For example, if we are using mitochondrial donation or pronuclear transfer, which is kind of oocyte donation, and also conventional oocyte donation when the oocyte is coming from the donor. So the most important part, which consists genetic material, this is a nucleus. But also we need to remember about cytoplasm, and we are learning more and more about organelles and the structure of the cells. And mitochondrials are playing really crucial role for the genetic inheritance and for creation of the embryo. So they are located in the cytoplasm. We cannot see them here on that picture, but we will talk a lot about them. And those cells, as zona pellucida, first polar body, corona radiata, or follicular cells, they are important in terms of the organelles and guarantee the safety of the creation of the cell and the development. However, they do not have a genetic material. So we need to remember that the genetic material we have in nucleus and in the cytoplasm, meaning in mitochondria, which are located in the cytoplasm. And if we are talking about human sperm, so we have a head with the nucleus inside, and this is that part which is used for ICSI. We have mitochondrion, which is also located in the midpiece of the spermatozoa. However, this part of the cell is not used for the fertilization. It can be used for the conventional IVF. However, through the science which we know already, it doesn't take part in the fertilization. And tail usually is not used because we are using for ICSI untailed spermatozoa. So what is inside of the cell? Inside of the cell, in the nucleus, we have chromosomes, which are created from the DNA, and the DNA consists from the genes. And 99.9% of the DNA of human beings are identical. So for all of us, even we all are different, 99.9% of our DNA is the same. And only 0.1% difference is known to make the difference in personal physical characteristics such as appearance, height, and constitution, as well as the development of the disease. So what is DNA? DNA is the chemical inside of the nucleus of the cell that carries the genetic instruction for making living organisms. This is material of the cell which carries genetic information, and the scientific name is deoxyribonucleic acid. The chromosomes are the packages of genes and other DNA in the nucleus of the cell. Chromosome contains hundreds and thousands of genes. Humans have 23 pairs of chromosomes, 46 in all, and 44 are autosomes, which codes different genes and also diseases, mutations, and presence of the individual. And 2 are sex chromosomes, which are responsible for, of course, male or female fetus. And each parent contributes one chromosome to each pair. So children get half of their chromosome from their mother and half from their father. Chromosomes are long stretches of DNA within which lie genes. A single chromosome may contain hundreds of genes. And for some genes, we know already their location as well as their coding. You probably know that scientists are working right now for full genome sequencing, however, and we have a lot of developments there. However, full genome is not read yet. And every day, every week, every month, we are learning more and more about this. So gene is the functional and physical unit of the heredity passed from the parent to the offspring. Gene is a section of the DNA which carries the information for a particular trait or characteristics. If we are talking about how baby will look like, so we may talk about phenotypical characteristics first, such as eye color, hair color, also production of particular enzymes, and also specific characteristics and diseases. A trait is any gene-determined characteristics, and it's often determined by more than one gene. Alleles are different forms of the same gene. For example, if we are talking about eye color, so there is one gene, however, it can have several alleles, and these alleles are responsible for blue, brown, and green or gray color of the eyes. Full genome sequencing is also based on a DNA molecular, and as I said, we are developing in this a lot right now. Proteins are also important to be mentioned if we are talking about oxygenation and genetics. Proteins are the most important class of the material in our body. They are not just building blocks for muscles and connective tissue and skin and other structures, but they are also needed to make enzymes. And enzymes are a complex protein that control and carry out nearly all chemical processes and reactions in our body. The body produces thousands of different enzymes, and the entire structure and function of the body is governed by the types and amount of proteins and the body's synthesis. Protein synthesis is controlled by genes, which also contained on chromosomes. So meaning our DNA is also based on the protein's production. And if we are talking about DNA, there is another class of genetic material which is not DNA itself, but this is RNA. It's ribonucleic acid. It differs to DNA by the lack of one oxygen atom, and RNA is involved in different cell processes such as protein synthesis, meaning is like a pre-DNA. And if you are talking about oxygenation, And if DNA is coming from the nucleus, so RNA will be present in the mother's body, and RNA will be synthesized by the body of the mother, who is carrying the genetic material from the donor and the father. And still, we don't have a lot of information from here, but many scientists, they are sure about impact of RNA also on the DNA function and the production and coding. And probably we will get more information about it in the future. Another interesting part of our genome is mitochondrial chromosomes. So mitochondria are the tiny structures inside of the cell, as I mentioned in the beginning, that synthesize molecules used for energy. So mitochondria is responsible for energy production for the cell, and it also gives a power for development. And this development is the most important for embryo production if we are talking about IVF. Unlike other structures inside the cell, each mitochondrial contains its own circular chromosome. So we may talk about mitochondrial DNA. This chromosome contains DNA, which we call mitochondrial DNA, that codes for some, but not all, of the proteins that make up that mitochondria. And mitochondrial DNA usually comes only from the person's mother, because in general, when egg is fertilized, only mitochondria from an egg become part of developing embryo. And mitochondria from the sperm usually do not become part of the developing embryo, and we may see the mitochondria, the round chromosome, and also parts of genes which are detected and connected to some diseases such as Parkinson, dystonia, ataxia, etc. So also we have treatment which we already call mitochondrial treatment, but also we are working more and more about mitochondrial donation, which gives a possibility for the intended parents to use structure of DNA from the central DNA, from the nucleus, and also DNA from mitochondria. If we are talking about the phenotypical signs of a baby, so the color, for example, of the future child's eyes, even if parents will be carriers of the genes which phenotypically product as brown eyes or dark eyes, this doesn't say for 100% that the child will have also dark eyes. As you will see the upper line here, so both parents are having dark eyes. However, the probability for child carrying the dark eyes will be just 75%, and there will be a probability of having green and blue as well. So why it is happening and how can we predict this and how this works? We have also strong and big genes. So modern science can predict that physical characteristics of a baby which inherit from mother and the father. And all this information is encoded into the genes, which we divide into strong and big. For example, strong genes are dark skin, dark colored eyes, curly and dark hair, big nose, thick lips, white chin, etc. These are just those genes which are related to the phenotypical signs. And the big genes is red and light color hair, straight hair, gray and blue eyes, light skin, and left-handed for the children. So in those pictures, you can see how genes are working. If you're talking about strong genes like brunette and curly hair, it comes from the mother on that pictures. However, brown eyes are coming from the father to their child. Another example, red hair is not coming to the boy, but instead from the father we have a brown hair and a big nose as straight genes. Again, dark skin and dark eyes and thick lips are from some from the father and mother. But also we need to remember that person's physical characteristics are the result of the displacement of several genes of their parents. So, and exactly, we cannot always predict how they will be divided, and certainly how the baby will look like, because genes can even skip several generations and then reappear, meaning that person can inherit certain genes from their grandparents or great-grandparents. That's why it is important to select phenotypical characteristics if we are talking about oocyte donation. And for oocyte donation programs, all these characteristics are available. And egg donation, this is the most effective way of getting pregnant for those patients who have problems with their oocyte production, or for older patients, advanced maternal age, patients after chemotherapy, or reduced ovarian reserve, etc. So in any age, oocyte donation is the best treatment. And if we are talking about donor selection criteria based on the knowledges which we just received about genetics, there are two important characteristics which usually doctor recommends to take care of. And also this depends on if you're going to tell your baby that he or she was born due to the usage of oocyte donor or not. So if you decide not to tell a baby about oocyte donation, you need to think about blood type of yourself and your parent in order to perform matching. That child will get a matched blood type of you, your donor, and your parent. And you can see on the picture that, for example, if you and your parent have blood type O, so you need to select a donor only with the blood type O, because the probability of getting another blood type is 0. So baby where mother and father has blood type O will get only blood type O. But if you are blood type A, for example, and your husband is blood type B, so child can get any blood type from the child. That's why you can select also a donor with any blood type. Usually it's easy to understand this structure, and if you need to consult, you just ask your physician once you're using oocyte donation. But again, it depends, are you going to say this to your baby or not? Phenotypical signs are important, as we understood already. However, we need to remember that 50% of the genome of a baby will come from your husband. So, and also based on the strong or weak genes, you will get a probability of those genes to be present in the baby phenotypical characteristics. But also, we still don't know, but those mitochondria and RNAs, they are playing quite important role. And that's why very often, as you can ask your friends or colleagues or any patients, once a baby is born using oocyte donation, all parents are saying that he or she is very much looking like me. Even if oocyte, this comes from the mothers, and even if oocyte donation was used. So still, probably we don't know everything, but there is something which also plays the role. Other factors such as education or background or any characteristics which is related to the character, we all understand that this is something what will be given to a baby once he or she will be developing in the family. And there were many examples of even identical twins being grown in different families and that they got different habits. Genetic match is also important, and it's considered now as a good clinical practice. We already can perform a lot of checking for different gene mutations, and this can be done also for your oocyte donor and for the parent, for the male partner. And if you will get this genetic match, you can avoid for a baby the fact that he or she will be carrier of the mutations. It's not an obligatory thing. However, it is, as I said, a good clinical practice and which can be offered by the clinics or by the oocyte banks. So based on these characteristics, you may select your donor or even embryo donation. You can also consider, but considering those few points. Usually banks or clinics are offering a database of wide characteristics, and what is important to be, this is wide personal profile, including all information about phenotypical signs, medical history, infection screening, genetic screening, consultations from the main physicians. And depending on the fact if oocyte donation is identifiable or anonymous, there would be a probability of having childhood photo or adult photo of the donor, so you can match different typical signs. Also, during the period of COVID, we know that there are many restrictions also for traveling outside of the country. So, for example, Origin Egg Bank is working for helping you to receive biological materials in your country. So if oocyte donation is possible in your country, your clinic can import biological materials for your treatment. And there are many successful cases like this. And if oocyte donation is not possible in your country, you can already travel to Ukraine to get it fully done here. And we spoke already about all these processes, and you can get back to the website of IVF Media Egg Donation Friends and find out more information. What we would like, I would like to present once again, this is the fact that for poor egg quality, and for older maternal age, mitochondria is playing a crucial role, and this is a battery of the cell. And if this mitochondria is older again, so the level of the activity is going down. And because this level of the activity is going down, this plays a role for the embryo creation and the embryo development. So we can get fertilization arrest, we can get lower quality embryos. That's why the treatment using pronuclear transfer or mitochondrial transfer, this is one of the successful treatments for advanced maternal age. And this treatment also will give you a probability of keeping your genome. We consider 98% of genome is coming from nucleus and 2% is coming from the mitochondria. And each couple have a social and genetic right to carry their own genes during pregnancy. So this is a great alternative for those who still may produce few of your own oocytes. However, the results of the treatment are inappropriate. And there will be a probability of using pronuclear transfer for creation of your embryos. So how it is happening? We fertilize both your oocyte and donor oocyte with your husband's sperm. And once we have two pronucleus, we are taking pronuclei with donor fertilized out, and we put them into the cytoplasm, including your mitochondria, which is helping to develop embryos better. So by this, you are mixing the genetic material and you have a probability to carrying your genes. There is no risk for fertilization because the transfer is already done between the zygotes. There is much better blastocyst outcomes. It will give probability for families affected by serious mitochondrial diseases a chance of having healthy children free of devastating and often limited disease. And the techniques and mechanical micromanipulation requires experience and special cultivation method during micromanipulations. However, we are very successful already in this, as I said. Another fact which I would like to talk about, this is the rate of genetic abnormalities, and we all know that this increases also with age. However, even in oocyte donors, there is a probability of getting oocytes which will be aneuploid, and this probability is up to 30%. So what we would recommend for best clinical practice in the oocyte donation, this is to perform oocyte NGS testing or NGS testing of the embryos before transferring them. So in order to increase the chances and maximize the chances for successful IVF treatment using oocyte donation. So by testing the polar body of the oocyte, we can be sure that this oocyte is euploid and has the best chances to create high-quality blastocyst. Because if we have high-quality blastocyst and great endometrium, well-prepared and responsive endometrium, this is when we will be the most successful. And the advantages of polar body NGS testing from the patient perspective is that the fact that the oocyte donation program mostly brings financial responsibilities for patients. And it's not the first IVF treatment which has happened already for that family mostly. And we would like to optimize and maximize the chances. So by this, we will definitely do that. So to conclude, the most effective treatment for patients in advanced age is oocyte donation. There are many new technologies which are still experimental so far, but they are effective and may be accepted widely in the future. We may predict how the baby will look like, however, not for 100% understanding everything how our genes works. Advanced genetic testing become more actively used and considered as a good clinical practice, which can help to avoid the mutations for the baby and predict their health. And our knowledge in genetics still does not cover all, and it's a point of interest for the scientists. So thank you so much. This is everything what I would like to touch during this webinar, and I will be happy to ask your questions now. I'm sorry, I see that sometimes the connection is getting lower, so just let me know if something wasn't unclear.
Thank you so much, Dr. Uliana. As always, it's brilliant to have you here. I hope that everyone did hear you, of course, correctly, because everything was okay. The video may be a little bit slower, but other than that, we were able to hear you, okay? So you can turn on your camera, at least for now, but let us know if anything happens and you cannot hear Dr. Uliana, okay? Okay, so right now we have one question, so let's have a look at it. And of course, this is time for your questions. We will start our Q&A session, so if you have any more, go ahead and type those in. And the question that we've got, the first one is, could you explain epigenetics?
Yeah, epigenetics, this is a kind of relatively new direction in the science, which study of how behaviors and the environment can cause the changes and affect the way how our genes work. This science cannot change the reading of DNA itself, but how our body reacts for the DNA. Still, there are many controversies on this, and it's better than the genetics because I am IVF specialist, so I'm not that deeply in epigenetics, so I would better not comment on this. However, if there is an interest, I can pass this question to our genetician, and she will be happy to explain.
Sure. Thank you so much indeed, of course, for your question. Definitely interesting one. And as Dr. Liana mentioned, if you wish, just get in touch with Dr. Liana. There is a link. I will send it in a second as well. And there is an option for you to get in touch with Dr. Liana. And one more thing, Dr. Liana, we can try with your camera on, okay? For now, we will be able to hear you. So let's try it. I will let you know if we cannot hear you, okay? Perfect.
Okay, yeah, because I couldn't hear you for some time, but that's fine. I'm back with the camera as
Okay.
well.
Right now you can hear me, so it's okay. I mean, I will let you know if something is not right, but now it's okay. All right, let's have a look. Next question is from Sarah. How do you source the egg donors?
Thanks, Sarah. So we are working with oocyte donation for many years already, and my experience started in 2006. And during all these years in our clinical facilities, we had a separate department, which we called oocyte donation department, where people worked specially for donor recruitment, stimulation, screening, and they keep all the records for oocyte donors, and all the processes are controlled by them. So they are within the clinical structure, starting from advertisement, as I said, their motivation. They keep contact with all our donors. They have great communication and relationships with them, and we really it's very valuable for us, everything our egg donors are doing. We take care about their health status, about their, as I said, motivation and screening and safety and everything like this. So they like to come to our facilities to be an egg donors. Also, we may provide more information and you can check our database. However, this is really a wide database of well-screened and very responsible and effective oocyte donors.
And once again, thank you so much for your question and of course for your help with that. All right, next question is up. Any chance of baby looking like me in an embryo donation?
Yes, of course, there is a chance as well. And you need just to follow the recommendation, as we mentioned, for selection of the donor of your oocytes and your sperm based on the phenotypical characteristics and based on your characteristics, including the blood group and all critical phenotypical signs. And again, once carrying the pregnancy, potentially RNA will play the role for the enzymes production, and still this process is not well explained. However, there is a chance that this baby will look more like you because of this process, because you will be carrying this embryo. However, also, it's probably a joke, but I think it will be very related to this topic, that once people are living longer together, they become very similar with each other. And also the habits and phenotypical characteristics and everything from your baby will be very similar. This is not like the decision of not using oocyte donation or embryo donation if there is no other chance medically to get a baby, in considering the fact how your baby will look, it's not appropriate. So you should not, there is much less risk that your baby will not look like you, not to make a decision to use oocyte donation or embryo donation. I'm not sure if I explained this correctly. However, I would recommend to use oocyte donation or embryo donation, and work with your physician for the recommendation of how to select the most appropriate donor, and don't think about this fact that this is a risk that the baby will not look like you. It will be your baby, and you will get the relationships with this baby and with your partner, and this will be the great achievements you can get.
Thank you so much for the clarification. Indeed, of course, for your reassurance, because this is definitely the thing that is very, very hard for all the patients. So thank you for the reassurance for sure. All right, let's have a look. Okay, Carrie has added there are two parts. I am 41, have had one failed PGTA, normal frozen embryo transfer, and in 2-week wait with my second frozen embryo transfer. As PGTA is normal, embryos are usually more successful. Could the failure be due to mitochondria?
No, if there is an issue for improper implantation for the genetically tested embryos, so you should look for the reason for non-developing embryo after being euploid and genetically healthy, that there could be something in your endometrium or in your uterus or your immune system that is not giving a chance for this embryo to implant properly or to develop properly. Usually what you need to consider, this would be implantation window. This is a screening which is done by several ways. Mostly this is electronic microscopy of the pinopodias being growing during the implantation window of the endometrium, or gene sequencing for also successful implantation. You can ask your doctor for this test, as well as the immune status and your immune response for the embryo, because if your immune system is very active, your embryo could be considered as an enemy for your body. And NK cells could play a crucial role not to give a right development for this. So some treatment as IVIG treatment or intralipids, etc., it may help you as well. So the point of implantation or antibody treatment like your thyroid antibodies, etc. So this is where you need to look for the reasons.
Thank you, Carrie, for sharing. And of course, thank you for your advice right here as well. Okay, next question is also ready. I'm 43. I have a lot of eggs, but I assume most of them are abnormal. Would it be possible with egg donors together with the new thing you told about the mitochondrial adoption and try to keep my DNA?
Yes, so pronuclear transfer or mitochondrial transfer, this is the treatment which we recommend those patients who still produce their own eggs, because we need to have a source of the genetic material coming from nucleus, mostly for these patients. And if you are producing your eggs independently, we may use those material in order to create your embryos. And specifically for your case, I think it would be a great decision for using mitochondrial transfer or pronuclear transfer, and you will successfully keep your nuclear DNA for creation of your embryos.
Thank you once more for your answer to that question. Let's have a look. Next question is also ready. So I was going down donor route, but got scared about resentment the child may have. Having read some stuff, I don't know what to do now. Anything you can advise?
So Jackie, I can just advise you to talk more with your clinician regarding how oocyte donors are selected, how they are tested, how they are screened. You can ask for consultation of psychologists for your donor. For example, we do for all of our donors, and we are reducing all risks connected to the risk of any diseases, also psychological, and also controlling their motivation. So all our donors, they are adult and mature persons who understand their responsibility being a donor. And even they are doing this to get financial motivation. However, we call this just a motivation for also for help, because for most of them, they definitely, or all of them, they feel their responsibility and the fact what they are doing a great thing for the patient, as definitely they do. So you need to communicate more with your clinical facility not to get your scares out, and continue, of course, with oocyte donation if this is the only way for you to get pregnant.
Thank you, Dr. Uliana, for that advice once again. And of course, Jackie, let me just mention that tonight we have this video done at 8 p.m. UK time. There's another webinar, and we will actually discuss emotional aspects of donor conception. So if you have time tonight, I can only invite you to join us, and maybe you can also ask your concerns, share your concerns. And of course, Becky Kearns, she's actually she did go through egg donation. She may have some advice for you as well. Okay, so thank you so much indeed. Let's have a look. There is one more question and actually comment. I have been told by a friend recently that 40% of the expectant mother's DNA goes into a baby when they are carrying it. I thought this webinar might be addressing this question. Anything you can add?
It's not about DNA, because we just explained that DNA comes from the nucleus, from the donor, and from the mitochondrial DNA. This is all we currently know about DNA. So this information can be connected to the RNA activities and the enzyme production, because enzymes, they can change the reading of the DNA. This is what we were talking. We can address this to the first question about the epigenetics. However, about this information for 40%, it's not confirmed yet. So it's an assumption, or this is what we may predict or we may think about. But for example, if we are checking genome and the genes and the mutations and carriers of the alleles, etc., for the babies which are born using the oocyte donation, they keep genome of donor and the father. So it still comes from there. But in the creation of a new life, there is newly created mutations and newly created pairs and nuclear components. So still, there are a lot of discussion about that the fact that once carrying the pregnancy, even with the genetic material of a donor and the father, mothers, more likely RNA plays the role into the genetic status of the baby. However, it is not confirmed yet.
Wonderful. Thank you so much. Once again, of course, for yet another question, comment, but also for your answer. And at least for now, it might be our final question, but of course, if you have any left, you know what to do. Go ahead, type those in. And Sarah has added, is it the case that sometimes a particular donor would not be a good match and lead to a failed cycle, but another donor would work? Is this common?
Great question. Thank you so much for this. Actually, donors, they are also individuals, and only clinical base, which is providing oocyte donation, either this is a clinic or this is an oocyte bank, can do their best in order to select so-called proven donors. For example, like after we perform the selection of the donor based on the selection criteria, inclusion criteria in terms of the age and phenotypical signs, and after initial screening is performed, after psychologists did the consultation and geneticists did the consultation, we are selecting 3 out of 10 candidates. However, after we are performing first stimulation route, and we are getting the outcome of the treatment for this donor, for some donors, we can also say, no, thank you, we will not go and continue with you being a donor. Because we are doing a lot of the selection process also for donor material, not just for her as being an adult organism, let's say, but also on her oocytes being produced. We categorize oocyte into 3 categories, Q1, Q2, and Q3. And if donor produces mostly Q2 or Q3 categories of oocyte, we will not continue using that donor in the stimulation cycles for the future. Also, getting more information about using the donor oocyte in different cycles, we can have cumulative result of blastulation and clinical pregnancies. So some donors, we call them so-called VIP and proven donors, they have great blastulation, they have great clinical outcomes, and some, they don't have the same. They have relatively lower. So we also, based on the clinical outcomes, we exclude those donors from the database. You will ask, how can we predict this from the very beginning? So we do a lot in comparison to some different clinics by checking the morphology and by offering also NGS testing, what I've just mentioned. So we can offer only euploid, meaning genetically healthy oocytes, but again, this is on demand on the patient. And we keep our database of outcomes from the donor cycles, and we always can offer proven donor for the treatment cycle if this is needed, of course. Also, there are tissue components. Like now we are developing a new protocol for the screening and selection based on the tissue. These are KIR receptors or HLA receptors, meaning that we can help to select a donor based on the tissue synchronization or tissue that this donor will fit your organism as much as the donor for donor organs. So this is something new also, which we are not commonly used right now, because even without this, oocyte donation is very successful in our center and using the materials from our bank. However, this is still a next step. This is still how can we improve this.
Excellent. Thank you so much once again. Interesting question indeed. So thank you. And of course, as always, great answer. Okay, let's have a look. There is another question. In February, I donated 2 embryos and failed. The endometrium was great and I had good health findings. I have no disease, but I have hypogonadotropin hypogonadism, and I had 10 IVFs to last always with my eggs. Is it up to the uterus? What would you advise me?
Still there are a lot of information which we would need to clarify. First, what was the quality of those embryos? Either they were blastocysts or not. I assume they were blastocysts, but then we would need to ask for the quality of those blastocysts just based on the morphological reading. Like what was the grade of the blastocyst and the quality. However, also the fact that if they were not tested for their genetic status, unfortunately, some embryos even come from a site of a donor, they can be aneuploid, and aneuploid embryos will not be successfully implanting. So I would recommend to use or to offer you the NGS testing of the embryos. However, grade endometrium, this is not much to say. So we need to know the thickness of this endometrium. We need to check the probability of your implantation window. This is what I mentioned, but about 11% of women, they have moved window of implantation, meaning in natural cycles, you can get pregnant because the embryo is counting and they talk with the endometrium when is the best time for implantation. And the smooth window of implantation can be moved either to the beginning of the cycle, meaning usually we transfer embryos on day 6, like 6 days of progesterone, and we transfer blastocysts which have 5 days of their development. So this is the routine practice everywhere. However, 11% of the patient, they will not have their endometrium ready for implantation in that time. And embryo is live inside of our uterus up to 48 hours. And if the implantation window is moved either for the beginning or later, within the period during 2 days, we can just transfer the embryos. However, endometrium is not ready, they will have no chance to implant. And once the window of implantation will be open, the embryo will not be alive there. So that's why we need to check the implantation window of potential, specifically for patients like you with 10 IVF attempts previously. If you had the same transfer in the same day, on day 6 of your progesterone. So maybe something will be there. Your hypogonadotropic hypogonadism probably was covered by the hormones. So your estrogen level should be fine if you had a medicated cycle prepared for your embryo transfer. However, thickness of endometrium should be over 7 millimeters. This is something what is optimal for successful implantation. With less thickness of endometrium, the probability of successful implantation going down. So here I would recommend to test embryos for making sure they are genetically healthy, and to consider testing your window of implantation, having so many failed cycles before.
And as always, again, a thorough answer. And as you can see, Zana would like to thank you for that. Thank you indeed. Okay, next question from Sylvia, a shorter one. How many times a donor can donate her eggs?
In Ukraine, there is a recommendation to donate not more than 8 times. And this is what we keep also as a rule for our donors. However, from the practical experience, we don't see any changes between the quality of the oocytes being produced in the first in comparison to the, for example, 6 cycle of a donor, if this is a good donor, if she's a good responder. Because we are explaining this process also to our donors and to all our patients, that every month we are getting certain number of follicles which are ready to grow. These all are follicles which we can see during the ultrasound scan, all small ones over 2 millimeters. And depending on in which age we are, a certain number is available. For oocyte donors, this is usually between 10 to 20 in each ovary. And only because hypophysis is producing a small certain amount of FSH, which is enough to grow 1 follicle, we are producing 1 dominant follicle in that cycle. However, all others, they will disappear. So they never will be used again. So 1 month, we receive 10 to 20 follicles in our ovaries, 1 developed to become dominant and ovulated, but all others, they came into the process of apoptosis. They just disappeared. So what we are doing once we are performing IVF cycle, we are just helping with additional dosage of FSH to grow all those follicles which are ready to grow, which are sensitive to exogenous or endogenous FSH. And we are not decreasing the potential of a new cycle to recruit new follicles, because new cycle, this is absolutely new page for us as women, and new cohort of the oocyte will be produced again and ready for FSH stimulation. So that's why there is no difference and no harm for the oocyte donors, as well for the patients during their IVF stimulation.
Excellent. Many, many thanks. Once again, another thorough answer for sure. Someone is typing, so let's have a look. There's a thank you from Sylvia. And it looks like that might have been our final question. But of course, as always, I want to mention that if you have any questions, not even now, but later on, anything that you might think about, you can always get in touch with Dr. Liana, and I'm sure she will be happy to help you. She and her team at I've Met. There is a link. I've sent this a bit earlier, so you can use that. And if not, we will be finishing for tonight, of course. But thank you so much, everyone, for your amazing question, for sharing also your stories, because we know that it's difficult and tough for you. So thank you for being here with us. And of course, we are happy that we are able to help you, even if a little. Dr. Liana, as you can see, there are thank yous coming up your way. So before we finish, is there anything at all you would like to add?
Thank you so much, you all, and this is as usual my pleasure to join you during these webinars. And I would like to offer all our patients not to consider this long. If you have a thought to go ahead with any kind of your IVF treatment, please go, because the time is very crucial. And for us, for clinicians, it's usually great to have you in your treatment in your young age, in your good responding time. However, if this is not possible in any time when you decided, and we'll be happy to coordinate and to help in pregnancy support and getting that pregnancy possible and treat you. So thank you so much, and do not consider long. Bye-bye.
Thank you so much indeed, Dr. Liana. It's always a pleasure. And let me just mention that actually Dr. Liana will be back next week. We will have another webinar with her. So I'm already looking forward to it. We will talk about ovarian aging. So I can only encourage you to join us. As you can imagine, it will be interesting for sure. And again, you will have a chance to ask your questions. And as you can see, more of those thank yous are coming up indeed. Well, I can only advise you to join us tonight. As I've mentioned, there is another webinar at 8 p.m. UK time. So very soon, we will also discuss egg donation, but more emotional aspect of it. So if you are free, join us. And I'm sure it's going to be interesting for you as well. Thank you. Dr. Liana, see you next week. And of course, everyone, have a lovely evening. And I hope to see you soon
Thank you.
here.
Bye.
Thank you so much. Bye.